I'm Ignacio Vela. I'm an associate professor at the Complutense University of Madrid, and currently I'm PI at the Microbial Interactions and Ecology Lab in this university. And in my group, we are interested in understanding the ecological and molecular mechanisms of interspecies interactions, especially in microbial ecosystems of industrial interest. So today I'm going to present some of our recent work in wine microbial communities, trying to show how, at least for me, wine fermentations can be used as model systems in community ecology. So, as you all may know, wine production is intimately associated with microbes, as they play key roles in every single step of the winemaking process, from their role in the vineyard as determinants of the health and nutritional status of grapevines, to, of course, their well-known role as conductors of the wine fermentation in the winery. So during the last 10 years, I had the opportunity to work in studying microbial communities throughout the entire winemaking process, from the vineyard soil to the grape mast and later the wine fermentation, and even studying the impact of wine fermentation in the gut microbiome of moderate wine consumers. So I think that I have a broad picture of the wine microbiome, and today's talk, I will try to show you some insights about microbiome patterns in wine fermentation ecosystems. So I want to start presenting some of the reasons why I find the wine as a very unique and interesting model system in microbial ecology. So first is because it has allowed us to work at different spatial scales, from the global scale to the regional and local scale. But in addition, this system allows us to address different biological questions at different levels of complexity, from the ecosystem and community levels to also levels of organism and genomes. And finally, as you will know during today's lecture, its unique balance between a moderate complexity and a full tractability allows to easily transfer knowledge from this system to other systems in similar agroecosystems, other microbial-based industrial processes, such as wastewater treatment plants, or even to propose new approaches and research frameworks in broader fields, such as theoretical ecology or systems biology. So I want to start presenting some insights on the wine microbiome, but now with a brief overview of the main results of this global survey, where we describe microbiome patterns in vineyard soils all around the world. So it was mainly descriptive work due to the limitations of the dataset that we had at this time, but it lays the necessary background for some current efforts. I want to highlight a current project led by my colleague Raúl Ochoa at the University of Cádiz here in Spain, from where we expect to provide a more comprehensive view of the vineyard microbiome patterns in very soon, in the next months or years. But let's back to 2020, when our collaborative effort with the company Biomakers and the group of Lars Hansen at Copenhagen University led us to building a wide dataset containing 250 soil samples collected from 200 vineyard blocks in 14 countries of the 5 continents where grapevines are cultivated, where we studied the composition and structure of bacterial and fungal communities by a classical 16S ITS amplicon sequencing approach. So very briefly, in these Venn diagrams, in particular, if you look into the very central section of the diagrams, you can see that the number of taxa that are collapsed at the genus level in this case, that can be considered as cosmopolitan taxa that defines the core fraction of the biomicrobiome, as we found them in most samples from all the 5 continents surveyed. And as you can see, this conserved fraction is higher in the bacterial than in fungal communities. In contrast, as you can see in the very external parts of this Venn diagram, the region-specific fraction of microbial communities is higher in the fungal than in bacterial communities. In fact, some climatic factors, such as temperature or rainfall, appear as better predictors of fungal alpha diversity patterns than are bacterial diversity patterns. In any case, as you can see here in this ordination plot, where each dot represents a soil sample and its position in the plot is determined by its microbiome composition. So 2 samples will be closer located as they have more similar microbial communities. So the biogeographical patterns found were strong at all spatial scales that we explored, from the global to the regional scale. since those samples coming from the same or closely located regions appear as clustered in this plot. So the nice thing here is that this strong biogeographical pattern allows us to fit predictive models that were able to predict the geographical origin of any new sample coming to the lab that was never used before to fit the model, just attending to the composition and structure of their microbial communities. So we can conclude that there is a microbial signature of the geographical origin in vineyards. So after describing and concluding these strong biogeographical patterns, we wonder how the farming practices applied in the vineyard may shape the fungal communities from the vineyard soil. So to answer this question, we use another data set, including a deeper sampling in two separate regions, Spain and the west coast of the US, as you can see from California to Oregon. And we collected a total of 175 soil samples from each country. And we annotated if that vineyards were managed under conventional, organic, or biodynamic farming practices. So for those of you that are not familiar with the term biodynamic in farming stuff, it is basically an extreme case of organic farming with certain almost esoteric practices, but that serve as a positive control of extreme organic practices, okay? So although we were looking for conserved patterns across regions, due to the strong biogeographical effect on beta diversity patterns, we analyzed the effect of farming practices in both Spain and US datasets separately, okay? So as you can see in this ordination plot, especially in the case of US sample, we detect a clustering pattern of samples depending on the farming practices applied in the vineyard, with conventional and biodynamic soil showing opposite diversity patterns, and those coming from organic vineyards in an intermediate position, as you can see in the plot. In Spain, we detected something similar about the contrasting behavior of communities from conventional and biodynamic vineyards, but organic vineyards showed quite similar diversity patterns to those observed in conventional vineyards. I have no time today to further discuss this specific result, but there are some potential explanations. So in any case, we went further in exploring the structure of fungal communities from these samples. So we were able to develop a new methodology based on co-occurrence networks, but now allowing to explore the structure of local communities. So using this methodology, we found a conserved pattern across the two regions regarding the topology of such local networks, with communities coming from conventional vineyards presented as highly modular and low cluster networks, while biodynamic samples were showing opposite features in both countries. So just to show you an actual image of local networks that are representative of the structure of fungal communities from these kind of farming systems, here you can see how low intervention practices, I mean, those from organic to biodynamic farming vineyards, led to highly clustered communities with strong association patterns between their constituent taxa. So just after concluding that the role of the winemaker and the vineyard farmer may have an impact in the diversity patterns of the vineyard soils, we wonder how, or if it had something to do with the fungal communities found in grape masts that are produced in the vineyard. So now we reduced the scale of sampling, and we now collected a total of 18 grape samples from 9 organic and 9 conventional vineyards located in 5 wine regions across Spain. And as you can see here, we detected again a strong biogeographical pattern in this ordination plot, but also a significant effect of the interaction between the origin and the farming practices applied in the vineyard. So we conclude that the farming practices applied in the vineyard not only affects the soil microbiome, but also the fungal communities that arrives to the winery in the form of the grape mast fungal communities. So just finally, in addition, we found a strong effect of the farming practices on the alpha diversity patterns of fungal communities in grape masts, with organic vineyards showing a clear higher diversity than conventional vineyards across all regions. So now let me introduce you. We're moving now from the vineyard to the winery or to the grape mass fermentation. So now let me introduce you some basic insights about the microbial diversity of wine fermentations using some results from a recent work from our lab where we analyzed the composition and structure of fungal communities of more than 270 samples coming from wineries from 6 different countries. And that allow us to build a catalog of more than 240 fungal genera that are detectable in wine samples by ITS amplicon sequencing. So approximately 80 of these genera have been already isolated in different labs all around the world. So we can have some metabolic information to anticipate or to unveil their potential contribution to wine fermentation. So our work and the work of many other colleagues have So our work and the work of many colleagues have concluded how the composition and the structure of fungal communities in wines is determined by the interaction of both biotic, abiotic, and anthropic factors, such as the biogeography, climate, and the type of management of the vineyard, the composition of the grape mass, the fermentation condition, and the addition of yeast inobula. And what interests us the most is the presence of interspecies interactions. So as you all may know, Saccharomyces cerevisiae is almost always responsible for completing the wine fermentation, but other non-Saccharomyces yeast have the potential to impact directly or indirectly, as we will see in this seminar, in wine fermentation performance. So in this work, we describe the main association patterns of Saccharomyces with the rest of the fungal genera in the most prevalent fraction of wine fungal microbiome, and found that although Saccharomyces cerevisiae and its main partners are not the dominant fraction of fungal community in fresh grape mass, they become dominant as the fermentation occurs, as this fraction obviously contains several moderately to highly fermentative yeast genera. Just to put some names in the table, of course, Saccharomyces, but you should be familiar to some other yeast genera, commonly known as non-Saccharomyces yeast genera, that are relevant for the winemaking process. As you can see here, we can highlight some of them, such as Strulaspora, Lachantia, Metzenicobia, Candida, Cryptococcus, Hansenaspora, etc. So, from an ecological point of view, a wine fermentation is a microbial ecological succession where the initial yeast community is mainly determined by the great osmotic pressure and the low pH of grape mast. And then it describes a progressive loss of diversity that is determined by the increasing concentration of ethanol and the decreasing availability of nutrients in the grape mast. That leads in most cases to the dominance of Saccharomyces cerevisiae. So in our lab, we are interested in studying what's the role of microbial interactions in all this process and how all these factors interact to finally define the most important thing in wine fermentation, that is the flavor of the resulting wines. So our main aims with this system is to explore and exploit the phenotypic diversity of wine yeast species and to understand the importance of microbial interactions in wine fermentations. So let's go with some of the results in this system, just telling you that since the beginning of this research line, about 10 years ago, we started, or we have tried to isolate wine yeast from different locations all around the world and from different grape varieties, different type of vineyards, leading to the establishment of a broad collection of more than 1,000 isolates at this moment that belongs to 30 different species. So assuming that the presence of different yeast in a grape mass allow to modulate the final sensory profile of the resulting wines, we have studied the potential of our yeast collection in order to improve both technological and sensorial aspects of wine fermentations. So I want to start by sharing some of the results of a recent work where we select a subset of 60 strains from our collection spanning the expectable phylogenetic diversity in wine yeast community. And we perform an exhaustive phenotyping of all these strains by measuring their growth phenotype in a panel of 28 fermentation conditions. to infer their environmental preferences. And then we measure their contribution to the metabolite profile of wines in laboratory scale fermentation using synthetic grape must as a media. Okay. So when we analyze the results of this phenotyping and connect them with the phylogenetic relationship of the strains in our collection, we find that there is a strong phylogenetic signal in wine yeast, as shown in this plot, that correlates the phylogenetic and the phenotypic distance between every pair of strains. So the existence of this phylogenetic signal allows us to fit models that are able to predict the phenotype of any new strain, both pertaining to a previously analyzed species or even to new species, based on the phylogenetic distance to another previously characterized strain from our collection. And as you can see here, it works pretty well with for almost all the 28 traits measured, except for the malic acid consumption capacity. So here you can see as an example of predicted versus observed efficiency of growth in synthetic grape must. And as you can see, the prediction power is quite high. Okay. So at this point, we wonder how we can use this knowledge, our collection, and this metabolic knowledge for the targeted improvement of wine flavor. So we focused on the production of hydrolytic enzymes as traits from our yeast collection to directly improve the production of wine aroma impacting compounds. So as you can see here in this plot, where I'm showing you the prevalence of 8 different enzymatic activities that are directly related with the aroma profile of wines, you can see that there are some highly prevalent activities, such as the beta-glucosidase or the protease, while there are some other enzymatic activities that are just restricted to a very limited number of species or even to a limited number of strains, such as the arabinofuranosidase or the beta-alliase, as you can see in the middle of the plot. So from now to the next 2-3 slides, we're going to focus on this specific enzymatic activity, the beta-alliase activity of yeast that are responsible for the production of volatile thiols. Volatile thiols are a couple of compounds that defines the varietal part of the varietal fraction of wines. And we can highlight these compounds because of its very low sensory threshold that are in parts per billion, okay? 60 nanograms per liter and 0.8 nanograms per liter in the case of the 4-MSP product. And these compounds are responsible for the presence of some pleasant aromas, such as the presence of passion fruit, box tree, and grapefruit aromas especially in white wines. So just to understand the basics of thiols metabolism in wine yeast, we should know that these compounds are found in the form of non-volatile precursors in the grape that are usually bound to amino acids. So these precursors enter the cell through oligopeptide transporters, and once inside the cell, at least for the main compound, the 4-MSP, there is a single enzyme, the beta-alliase encoded by the gene IRC7, the main responsible for breaking the carbon-sulfur bond, allowing the yeast to consume the cysteine from this precursor and releasing the volatile fraction of the thiol that can be now perceived as part of the wine aroma. So it is worth mentioning that both the precursors, the transport inside the cell, and the beta-alliase activity, the intracellular beta-alliase activity, are both under the control of nitrogen catabolite repression. So the presence of high doses of nitrogen in wine fermentation may inhibit these activities. Just keep this in mind for some future results discussion, okay? So coming back to the IRC7 gene that, as I mentioned, is the only gene responsible for this beta-alliase activity in Saccharomyces cerevisiae, we found that there are 2 allelic variants of IRC7 in Saccharomyces. One producing a full-length allele of this enzyme, coding for a 400 amino acids enzyme, and a truncated version, which results in a shorter enzyme of 340 amino acids. So in wine yeast, we can find the 3 possible genotypes combining these alleles in wine yeast. But we conclude that only those containing the full-length version of the enzyme have a maximum beta-lase activity and have the capacity to produce volatile thiols in wine fermentations. However, we find a really surprising result that after checking these allelic variants in more than 220 Saccharomyces cerevisiae wine strains, we found that almost 90% of the wine isolates have the truncated version of gene in homozygosis, while only 3% have the functional version of this enzyme in homozygosis. So this was really shocking for us. And these were bad news for the industry, as it means that you cannot use the majority of Saccharomyces strains to increase the volatile thiols in wine. So just to better understand this surprising result, we performed a genomic survey in a collection of about 280 Saccharomyces genomes, studying the prevalence of the IRC7 allelic variants in different phylogenetic clades. And we found that the truncated version of this gene is only found in some domesticated clades, such as those coming from bread, beer, or wine fermentation, but it is completely absent in the wild clades containing genomes from strains that were isolated in forests all around the world. So this was an interesting observation. So just trying to understand the reason why the truncated version of IRC7 seemed to be favored in wine strains, we compare the growth fitness of 10 Saccharomyces cerevisiae strains of each genotype in a panel of 28 growth conditions, and found that those strains containing the low functional version of IRC7, I mean, the truncated version of the gene, have on average higher growth rates in all growth fermentation conditions. And when performing competition experiments between homozygous strains for the full-length allele version of IRC7 and those homozygous strains for the truncated version of this gene, we found that the HS strain tend to win most competition when growing in both fermentative and non-fermentative substrates. So I do not have the time to further explain this study at this moment, but at the end of the day, we confirm that it's not a good idea to use Saccharomyces cerevisiae to improve the volatile thiol composition of wine, since most of the wine strains do not have the enzymatic capacity to release them from their precursors, and those who have the enzymatic capacity have a poor fermentation performance. So we decided to came back to our collection of non-Saccharomyces wine yeast and found this strain of Torulaspora delbrueckii that showed the maximum beta-lase activity from our collection. We found that all the strains of these species that we have in our collection have an orthologous gene with high homology with the full-length allele of the IRC7 in Saccharomyces. So we decided to perform some simple laboratory scale fermentations to compare the use of two strains of Saccharomyces, one homozygous for the truncated version of IRC7, those that we call HS, and another strain that is homozygous for the full-length allele of IRC7 that we call HF. And we assay these two strains as single inocula, and the use of these strains in co-inoculation with our Torulaspora delbrueckii strains. So looking at the, let me just show this plot. So looking at the yellow bar, the right bar, the very yellow bar in this plot, that corresponds to the concentration of our main volatile thiol of our interest, the 4-MSP. As expected, we do not detect 4-MSP in the fermentation carried out by the HS strain, but found a kind of detectable concentration of this compound just above the sensory threshold in the fermentations carried out by the HF strain. So the good news were that in the presence of our Torulaspora delbrueckii strains, the concentration of 4-MSP increased about 8 times. So this strain is now a commercial strain that is exploited by a yeast producing company. So since Torulaspora delbrueckii is not able to finish the fermentation by its own and should be always used as part of a mixed consortium with Saccharomyces cerevisiae, we decide to further study the interaction mechanisms between these two species. So we designed a pretty simple experiment in synthetic grape must, comparing the performance of Saccharomyces cerevisiae and Torulaspora delbrueckii as pure cultures and as part of the mixed culture with both species. And we assayed this single and mixed culture in 2 types of synthetic grape must, one containing a low concentration of nitrogen in the form of ammonium, and another one containing an extremely high concentration of ammonium to study the regulation of the nitrogen catabolite repression in this species that, as you can remember, it plays a crucial role in the metabolism of volatile thiols, right? So in this experiment, we analyze both the metabolite profile of the resulting wines and the transcriptional profile of Saccharomyces cerevisiae in the different conditions assayed, okay? So first, we found that the metabolite profiles of the resulting wines, as you can see in this plot, in the case of the metabolite profile found in the mixed inoculum, they were more similar to the profile of the wine produced just using Saccharomyces cerevisiae than to those produced just with Torulaspora. This is something expected since Saccharomyces will dominate the mixed fermentation, so it will contribute mostly to the metabolite profile of the resulting wines. We also found a conserved pattern effect of the nitrogen concentration in all the inoculation conditions, as can be seen here in the second principal component of the plot. But after a deeper look into this ordination plot, focusing on the Euclidean distance between the samples in the plot, we found that the presence of Torulaspora seemed to reduce the effect of the nitrogen in the final metabolite profile of the wines. So we go through the transcriptome data, and looking at the transcriptome of Saccharomyces, we demonstrated that the presence of Torulaspora delbrueckii, as shown by the purple circles here, and the purple bars in the upper part of the slide, was reducing the transcriptional impact of the extreme concentrations of nitrogen in Saccharomyces cerevisiae. So this result was key because we demonstrate that interspecific interactions appear now as a key factor in shaping the performance of wine yeast consortia. So when using a mixed inocula in wine fermentation, we cannot expect the result to be the sum of the metabolic potential of Saccharomyces plus the other non-Saccharomyces species, but we demonstrate that the non-Saccharomyces species have also an indirect effect by changing the way that Saccharomyces is acting or is performing the wine fermentation. So finally, we wonder if we can understand and ultimately predict the outcome of interspecies interaction in wine yeast consortia. So to answer this question, we select 10 non-Saccharomyces yeast species from our collection, and we constructed hundreds of synthetic yeast consortia as random combinations of 2 to 6 species, and assayed these consortia with and without the addition of 2 different strains of Saccharomyces cerevisiae. So then we assess their performance by measuring the main ecological function of wine yeast communities, that is their sugar consumption capacity, okay? So as you can see in this plot, those communities containing Saccharomyces cerevisiae, where we add Saccharomyces cerevisiae strains, consume a higher fraction of fermentable sugars than the corresponding background communities without Saccharomyces cerevisiae strains. In fact, in most cases, those communities that contains where we inoculate Saccharomyces cerevisiae were able to complete the wine fermentation, meaning that they consume more than 95% of fermentable sugars. The interesting point here is that since Saccharomyces cerevisiae alone is able to complete the consumption of all fermentable sugars in our experimental conditions, finding certain combinations of species where Saccharomyces cerevisiae is not able to complete the wine fermentation, as you can see here in this plot in orange, indicates the existence of negative interactions in some ecological context. So in this new plot, we are representing the average contribution of the different yeast species assayed in this trial to the function of the communities where they were at. So this plot allows us to identify some qualitative patterns on the contribution of species to wine fermentation, especially for those species with a pretty marked negative or positive contribution. As you can see here, for example, these species MP or PK that are Metzenicobea pulcherrima or Pichia kuldiapsevi have a marked negative contribution to the fermentation performance of complex yeast communities, while other species such as Lachansia thermotolerans, here marked as LT, and the two Saccharomyces cerevisiae strains, SC5 and SC8, have a very marked positive contribution independently on the community where they were at. Okay, so the challenge now was to be able not to qualitatively predict the impact of a species, but to quantitatively predict the performance of complex yeast consortia using this data, especially seeing that some species had not such a clear positive or negative effect, but kind of a context-dependent effect. So to be able to mathematically describe this context-dependent behavior, we had the pleasure to collaborate with the group of our colleague Álvaro Sánchez, at that time working at Yale University, now working at the Spanish Research Council here in Spain, in Salamanca. So his group developed this new framework that, mirroring the global epistasis phenomenon in genetics, proposed that the functional contribution to a species in complex communities depends on the function of the background community where it is set. So, as you can see in this new plot, that we're representing the differential function that is added by one species, in this case Saccharomyces, to the function of different background communities with different level of performance, from low functional to highly fermentative communities. In this case of non-Saccharomyces yeast, you can see these low to high fermentative communities from the left to the right in the x-axis. So a priori, the potential contribution of a species will be always higher in low functional background communities, right? As can be clearly seen here for Saccharomyces cerevisiae, that in the left part of this plot, the delta fraction of the function that Saccharomyces is adding to the function of the community is higher than the differential function that is adding Saccharomyces to the function of highly functioning communities. So now, looking to these plots for other species, the same occur with other similar non-Saccharomyces species, as you can see here in the left, in the case of Pterolastra delbrueckii. So contrary, as anticipated before in the previous plot, some species have always a negative effect on the functioning of fermentative yeast communities. But the interesting thing now is that we can clearly see how certain species, such as in this case hop, that is Hansenaspera opuntiae, have a clear context-dependent effect, having a positive contribution when added to low functional communities, but turning to a negative effect when they are added to a highly fermentative community. So the magic here is that using some basic attributes from this functional effect equation that are basically the slope and the intercept of this functional effect equation, we can define the ecological function of any species in different diversity contexts. So just putting together, concatenating the functional effect equations of those species that are contained in a given community, we can quantitatively predict its function. As you can see in this plot, where we show the predicted versus observed function of a new set of 131 new random combinations of species that were never assayed before. So you can see that the predictive model works pretty well. So we basically were able to predict the fermentative performance in terms of the fraction of sugars that will be consumed during the wine fermentation of any combinations of these 10 non-Saccharomyces species with different Saccharomyces cerevisiae strains. So we're really excited about this result, and we now wonder if we can predict not just this basic function, but more complex metabolic functions, such as the production of a specific aroma impact compounds or the fermentation performance of yeast consortia in suboptimal fermentation conditions. So just to finish my presentation, I just want to briefly mention some of the questions that we are currently working on once we have established the tools and the methodologies for using wine fermentations as tractable experimental systems, that they are mainly focused on the study of the molecular mechanisms that are under these interspecies interactions in wine yeast consortia. So one of the main questions that we are working at this moment is what are the metabolic niches and ecosystem functions of these non-Saccharomyces yeast species that appear during the first stages of the wine fermentation, but then disappear as Saccharomyces dominates the process. We believe that they play a relevant role in the ecosystem function. So we aim to identify the molecular determinants of the contribution of these species to be able to predict how they contribute from the molecular point of view, for example, to the aroma of wines. Okay. A couple of years ago, we present a kind of crazy idea in one paper where we propose to build a Frankenstein-inspired yeast where we introduce in a synthetic yeast a 17th chromosome containing the metabolic functions of all the other members of wine yeast consortia that are not encoded in the genome of Saccharomyces. So we can have like a whole ecosystem in a cell where Saccharomyces will be able to cover all the metabolic functions that are covered in complex wine yeast consortia. So this was a crazy idea that was developed with my colleague Saki Pretorius in Macquarie University in Sydney. And the other fundamental question that we are currently addressing is how conserved is the molecular response of Saccharomyces to interspecies interactions in different diversity contexts, and if it changes in different abiotic contexts. So this last part of the question leads us to kind of an applied aim. That is, if we are able to engineer from the top down the composition and function of complex yeast communities isolated from grape mast, through rationally manipulating interspecies interaction, that we will be able to change by modifying the environmental or the fermentative conditions. Since, for example, in the wine industry, you can add specific nitrogen nutrients, you can change the temperature of the fermentation, you can add more or less sulfide. You can have some power to manipulate the environment, and we are measuring how these typical enological practices are actually changing the way that wine yeast interacts. So that's all from my part. I want to thank you for this time listening to this talk, and I want to thank my collaborators that participate in all the work presented today. So thank you very much.
Thank you very much, Ignacio, for the beautiful presentation. And thanks everyone else for joining today's webinar and sending your questions. So we already have 2 questions in the Q&A, so I'll read that out. So the first question is, was the transcriptome in the mixed fermentations analyzed in bulk, or did you assign the transcriptome signal to each strain?
Not in this case, we obtain all the raw sequences from we sequence the entire transcriptome of the mixed cultures. We use the raw reads against a hybrid genome containing the genome of Saccharomyces and Lactantia, and we just analyzed the transcriptome of Saccharomyces. In this paper, you can see the different transcriptional regulation of Torulaspora and Saccharomyces when attending to the fraction of transcripts from one species and the other one. But in today's talk, I just, the insights that I shared today was just focused on Saccharomyces. But if the person who shared this question have a deeper interest, I can share the paper where he or she can see the different. I mean, and it was interesting since Saccharomyces and Torulaspora seems to have like kind of different response to nitrogen catabolite repression, and Saccharomyces appeared as to be more or to have a strong repression by ammonium than compared with Torulaspora.
Yeah. Okay, that's great. Thank you. Yeah, the person who asked about the transcriptome question, they have asked that, yeah, if it would be great if you could share the paper. So if you could send the paper to me, I'll include it in my email that I will send along with the slides.
What I can do is to share the 4 main papers that I used like to organize my presentation today, that are kind of representative from both the vineyard staff and the wine fermentation staff.
Great, that would be really helpful.
Okay.
Okay, so the next question is, I think it's about application. Do you have plans to work with the wine industry, or do you expect to apply this knowledge in the industry?
Yeah. In fact, my background was kind of the opposite that the typical career of researchers, since I came from a very applied research background. In fact, my PhD was fully funded by private companies that are not only wineries, but also these companies of the auxiliary sector, that the wine yeast producers, basically, that sell wine yeast strains worldwide. So yeah, at the beginning of my career, I worked really closely in collaboration with the industry. Now I kind of need some years to think about my own research line and to establish my group. So for the last 4 or 5 years, I've been mostly working in fundamental questions, just using public funding. And now I'm starting again to trying to see if these models and our yeast collection are of interest for the wine industry. And we're starting to collaborate with different companies, just trying to use this system to, I don't know, for example, in a particular case, for the design of a specific combinations of yeast species to achieve a specific metabolic goals, such as the production of a specific compound or the fermentation in a specific, like optimal fermentation kinetics in a specific suboptimal fermentation condition, for example.
Very good. Thank you. So yes, we have a new question just came in. Are there similar initiatives for producing wine with lower alcohol content?
Yeah, I mean, this is not my main area of expertise, but I think that there are 2 approaches. One of them involves the use of non-Saccharomyces, because non-Saccharomyces yeast species that almost always have a lower fermentative power than Saccharomyces can use sugars by respirative metabolism. So they can use to lower the concentration of ethanol in wines. The challenge here is not to increase a lot the concentration of acetic acid that could be an off flavor in wine that is associated with aerobic consumption of sugars. Okay, so this is one approach, and the other one is to modulate, for example, the Crabtree stuff in Saccharomyces. But it is kind of challenging. I mean, the best scenario using biological tools is to reduce 1-2% the ethanol concentration in wines. So to obtain like ethanol-free wines is just by physical procedures.
Great, thank you. So we don't have any new question yet. We still have some time. So if anyone has any other question, you can type them in. And while we are waiting for a new question, I would just like to tell you that, this webinar was part of a series called Exploring Microbial Ecosystems. So this was the last webinar of this series. So we have in total 10 webinars in this series. I'll share the link of all the webinars with you in my email with all the links where you can go to those webinar pages and find the recording. So you will find some more webinars which you might find interesting for your research.
Okay, so just some self-promotion. So if anyone is kind of interested in the future of for applying to kind of a postdoctoral European grant or whatever, I think that you have in the presentation my contact. So yeah, we are always happy to receive international researchers in the lab. So we think that the model system that we have established have the power to answer not only those questions that I already shared, but maybe some other questions in community ecology or even in microbial evolution that some guy in the audience may have in his or her head. So feel free to contact me if you are interested in any collaboration or whatever. I'm always happy to share our knowledge and our strange collection. So feel free to contact.
Yeah, that's very good. Yeah, and just to let everyone know that, I'll share the slides with you in my email as well, so you can find all the contacts and everything. So there's just new question came in. Does the diversity of yeast species in a vineyard have implications for the wine stability? And what are the implications for using indigenous versus commercial yeast in winemaking?
What do you mean with wine stability? I mean, the stability of the final product. I mean, avoiding any alteration in the bottle.
Yeah, mostly the long lastingness and the flavor profile.
Okay. So we can hypothesize that the higher the diversity, the higher the metabolisms that are acting in a fermentation. So the higher the diversity of metabolites at the end of the fermentation. We are, in fact, now with this that is ongoing work, we are actually trying to demonstrate because we are analyzing different communities of increasing diversity from 1, 2, 3, 4, and 6 species. And we are measuring the metatranscriptome of all this community. We're aiming to demonstrate if it is true that more species means a more diverse metatranscriptome in terms of the number of biological pathways that are active, and if we can relate different metabolic pathways with specific aroma impact compounds. So theoretically, yes, but I have not seen any paper currently published that demonstrate that the higher diversity in yeast, the higher complexity in wine aroma. And I don't know about the stability, in terms of for example, the protein haze that is a flavoring wine. For example, the presence of specific yeast have been demonstrated to be able to produce proteases enzymes during the fermentation that reduce the risk of protein haze, that is this problem in bottles. Okay, and just the final question about the indigenous versus commercial wine yeast. From the very enological point of view, I cannot affirm that one option is better than the other one. What is true is that the use of commercial yeast in a winery that is closely located to the vineyards, to the estate, reduce the yeast diversity in the vineyard. I mean, we perform a study in different vineyard blocks all around a winery that were using the same commercial strain for years, and we see that those vinegar blocks that are closely located to the winery have almost in all, I mean, all the isolates that we obtain from grape mast pertain to this very specific commercial yeast strain. So it definitely reduced the biodiversity of the surrounding ecosystems around the winery.
Yeah,
It's not negative from an enological point of view. It's not a better wine, but it have these ecological consequences in surrounding ecosystems.
I think in line with the same question, there's another comment. I think diversity for Japan region was not studied. Do you think that should help? I could see Japanese sake was considered for analysis, hence just curious about that.
Yeah, absolutely. In fact, 1 year ago, I was in Japan in a conference and I was visiting some vineyards in there. They were mainly using foreign varieties, European and American varieties. But I think that, of course, the study of completely different systems, completely different weathers and different locations, especially in case of islands, is really important and will be really interesting for these global surveys. I think that in the new global survey that I told you before, that is a huge project led by my colleague in Cádiz, they do not have any sample from Japan. So it will be really, really interesting to analyze this kind of samples. And talking about other beverages such as sake, other fermentative systems, yeah, it's definitely really interesting. In fact, there is a increasing community of researchers that are trying to establish different fermentative products as model systems in ecology. There is another example that is kefir, and there are really nice researchers all around the world working on kefir. And as in the case of sake, it involves not only yeast but also bacteria and filamentous fungi. So yeah, it's definitely an interesting model system also, such as, for example, cheese is the same.
Okay, so thanks a lot and bye for now.
Thank you.